Greetings! My name is Samara Kanji, and today I will be presenting to you my experimental project on demyelination in diffuse axonal injury and mild traumatic brain injury. To begin with, mild traumatic brain injury, or mild TBI for short, is an injury that is caused by the direct impact to the head or a blast injury, causing the acceleration or deceleration of the skull. In youth, a common cause for mild TBI is due to sports. These sports-related injuries can also occur more than once during one's sports career as a young person, impacting their adolescent brain development. Little is known about the effect of repeated mild traumatic brain injury in adolescence, as there have not been many studies done with animal subjects. Mild traumatic brain injury consists of two parts: the primary injury, which is what takes place during the time of the injury, and the secondary injury, which is what takes place seconds, minutes, or days after the primary injury. A common type of secondary injury is known as diffuse axonal injury. So, what is diffuse axonal injury? Diffuse axonal injury, or short, DAI, is when the rotational and shearing forces from the primary injury stretch or tear the axon. As a result of diffuse axonal injury, it can have a few effects, such as the disruption of the axonal transport. This disrupts the flow of proteins, organelles, and synaptic vesicles. One specific protein that participates in this is known as the neurofilament heavy chain protein. The neurofilament provides structural support to the axons, but undergoes cytoskeletal distortion as a result of diffuse axonal injury. Diffuse axonal injury can also cause some brain swelling. This is because the axons are a part of the neurons, and when there is damage there, this can cause overall brain swelling. However, this can go away in just a few days. And finally, axonal pathology leads to the demyelination around the axon. Speaking of myelin, what is myelin? Myelin is a white fatty lipid substance. The myelin basic protein, or the MBP, is what helps with the myelination around the axon, forming the myelin sheath to protect it. So, the myelin sheath serves as protection, but also, myelin is what makes up the white matter tracts. The largest white matter tract in our brain is called the corpus callosum. Myelin also provides nutrients to neurofibers, and it increases the speed of action potentials. These action potentials are the signals between the neurons which are used as communication. Now, when myelin is damaged or there is demyelination, the speed of the action potentials is a lot slower, and our neurons do not communicate with each other as fast enough. Current studies have shown that demyelination can occur after mild traumatic brain injury. However, there has not been many studies done on the effect of repeated mild traumatic brain injury in adolescence, which is an important time for the brain to develop. With that said, my research question is, does repeated mild traumatic brain injury in adolescence lead to demyelination and diffuse axonal injury? My hypothesis is, if the adolescent mice were to undergo repeated mild traumatic brain injury, then they will experience diffuse axonal injury and subsequently demyelination. Variables. For my independent variable, it was whether or not the mice would receive a mild traumatic brain injury or not. There were two groups, the sham mice, which were 6 mice that did not receive the injury, and the repeated mild traumatic brain injury mice, which were 6 mice that did receive these injuries. The dependent variable was the amount of myelin basic protein and the amount of neurofilament heavy chain protein in the images. My controlled variables were the age of the mice. They were all 48 postnatal days old. The sex of the mice, they were all male. The amount of anesthesia given to both groups of the mice per kilogram weight. So even the mice that did not receive an injury received anesthesia. The speed and weight of the cylindrical impact device, this was what was used to injure the mice. The amount of injuries received. So the mice that were supposed to receive an injury, they received 5 repeated mild traumatic brain injuries every 24 hours. The immunohistochemistry technique on the slices, and the technique used to image the slices. My confounding variables were the natural variability and the weight of the mice. Moving on to my procedure. Done under the ethical conditions at the University of Calgary, My first step was to injure the mice. After, we waited 5 days after they had received their final injury, and they were euthanized and their brains were taken and put into a cryostat, where they were sliced into 40 micrometer sections. I would also like to point out that I was not involved in the injuring, euthanization, or slicing of the mice brains. My involvement in this project came in the immunohistochemistry process, where we labeled the myelin basic protein and the neurofilament heavy chain protein in the images. I then mounted these slices onto slides and they were put into an automated confocal fluorescent microscope, also known as the Cell Discoverer 7. So, after imaging the slices, here are the images of the motor cortex. Since there were 6 mice in each group, we used 3 mice for the motor cortex and 3 mice for the corpus callosum. The 2 images on the left are the traumatic brain injury mouse, and the 2 images on the right are the sham mouse. The green fluorescent images are the neurofilament heavy chain proteins, and the red fluorescent images are the myelin basic protein fluorescent images. When I first took a look at these images, I did not see a significant difference in the amount of myelin in either of the images. Similarly, we wanted to look at this in the corpus callosum. The color coding is the same. The red images are the myelin basic protein images, and the green images are the neurofilament heavy chain protein images. Once we looked at this, we also saw that there was not a difference in the amount of myelin we saw in either one of the mice. We then wanted to quantify this. I would like to point out now that we were not able to quantify the images of the neurofilament heavy chain proteins because they were not imaged at a high enough resolution. However, I was able to get data from the myelin basic protein images. To quantify the myelin basic protein, the percentage area of the myelin basic protein fluorescent stains in the corpus callosum and the motor cortex were taken from all the mice. We then took their overall averages, and these dots here represent the different mice. Based on my first impression of the graph, I thought that the traumatic brain injury percentage area of the myelin basic protein was greater than the sham mouse. However, we took these values and conducted a t-test, and we found that our value was greater than 0.05, indicating that there was no statistical significant difference in the amount of myelin in either region of the brain. So, after analyzing the data, a conclusion that is made is contrary to our hypothesis, our results show that injured mice do not show a statistical significant difference in the amount of myelin or axonal loss compared to the sham mice. But why is that? Potential reasons are due to the limitations in our experiment, such as the sample size. We only had 3 mice and they were all male. Another point is the time post after the injuries had taken place. Between the time the mice had received their 5th and final injury and the time that their brains were euthanized, we waited 5 days. And finally, there could have been potential remyelination. This connects to my previous point, where between the time the mice had received their final injury and they were euthanized, there could have been potential remyelination in those 5 days, which is why we didn't see different results. So what will I be doing in the future? First off, I will image the neurofilament heavy chain images at a much higher resolution so that we can quantify their values. I will then increase the sample size and even include female mice. This is because another study done with mild traumatic brain injury showed that female patients are more prone to showing symptoms towards mild traumatic brain injury and have more abnormalities in their brain scans. Thirdly, I will observe the brain at a different time post. Rather than 5 days this time, I will observe the brain 1 day after the injury and then 24 hours after the injury. I will then observe if the injury has an effect later on during adulthood. So this means that we will injure the mice when they are in their adolescent and then look at them as adults to see if repeated mild traumatic brain injuries impacted the way they were as adults. Future applications. My project will contribute to the understanding of repeated mild-traumatic brain injury from adolescence to adulthood. It will add an understanding of molecular and cellular mechanisms behind repeated mild-traumatic brain injury, and it will help to develop better treatments and diagnostics. Acknowledgements. Special thanks to Dr. Alex Lohman and the Department of Biochemistry and Molecular Biology at the University of Calgary. I would also like to thank Thomas Carr for being there to answer all my questions, and Dr. Garcia for supporting me and guiding me throughout this process. Thank you so much for listening to my presentation.